physiologist with a research focus on modulation of neural plasticity, neuroregeneration, and neural repair by transcranial magnetic stimulations and magnetized nanoparticles in humans and animal models. Neurotraumatic disorders. Her dedication to research excellence is evident in her extensive publication work with over 100 peer reviewed articles and 575 citations in renowned scientific journals. A prominent figure like her is a recipient of multiple accolades, and to name a couple, our Ames Research Excellence Award and Dr. B.C. Roy National Award for Excellence in Healthcare, and many more. We are excited to have you join us, ma'am, and share your scientific wisdom, enrich our collective understanding. The title of our talk is Unlocking Neuromodulatory Role of External Magnetic Field Stimulation in the Rat Model and Neurodegenerative Disorders. Please welcome, mama.
Good morning, everyone. Uh, First of all, I would like to thank Professor B.N. Malik, Dr. Rachna, and the whole team for giving me this opportunity to interact with you all. The title of my talk is Neuromodulatory Role of Transcranial Magnetic Stimulation. So we have I being from a hospital setup, and therefore most of the time the work which I'm doing should have a translational value, and therefore you will find. So, therefore, we are trying. This is transcranial magnetic stimulation is one of the latest techniques, and it has just come up in last 10 to 15 years. So, before I go on, I'll just I got to know from Dr. Rachna that most of you are PhD students, and therefore I thought I'll start with the historical perspective that how this came into picture, right? So, Galvani, I hope all of you must have heard his name. Okay. So he was a physicist who was trying to, or a biologist who was trying to basically understand during that time that what is the effect of lightning. Now, generally, whenever there is rain, it is associated with lightning, and they wanted to understand that what is the effect of. Hi, good morning, Dr. Jha. No, I just started. So he was studying the effect of lightning, natural lightning on the body tissues, and therefore he was using frog as a model. And so, like always it happens, he had to study that, he had to remove the skin, and therefore the muscle tissue was exposed. And he had kept certain electrodes on the spinal cord. And he suddenly, and his assistant who was assisting, he was trying to remove the skin with the help of a scalpel, and scalpel got touched with the electrodes and muscles. There was a twitching in the muscles. Okay. So initially, obviously, they didn't, I mean, take things into perception. But then it's also again happened that the electrodes which were there, the free endings of that got touched with the iron rod. And then again, there was a twitching of the muscles. So when this happened repeatedly, so they thought that it's something is happening, and therefore they carefully planned the experiments on frog itself. And then they realized that whenever you are stimulating the electrodes onto the spinal cord, there's a twitching of muscle. And therefore he gave the concept of animal electricity. So that's how the story started. And then obviously a lot of experiments were performed. I'm just giving few key points. And it was around in 1835 that Faraday, he gave the concept of electromagnetic induction. All of us must have read this in our 12th class also, or physics also. And with a classical experiment that if you have coils, you pass the electricity through a coil of a wire and you place iron next to it, it gets magnetized, right? So that is what is electromagnetic induction. And therefore they realize that when the electricity is passed, then not only that the electricity per se can produce an effect, but the things can get magnetized, and therefore the magnetic fields are generated. Okay. Or a vice versa is also true, that if there is a changing magnetic field, that can induce electricity. And therefore now they tried to move, the scientists, they thought of whether magnetic fields itself, which is induced by electricity, can they stimulate the tissue? And therefore Magnuson, उसमें ऊपर से जो आ रहा है उसको हटा सकते हैं. So Magnuson and Stephen around 1896, so they were more interested to find out now not only in electrical signals or electricity per se, electrical current per se, but more about the magnetic fields. That magnetic fields which are generated, can they stimulate the nerve tissue or has certain effects on the body tissue? And therefore they devised, as you can see in that figure, lot of So they devised lot of big coils through which the electric current was passed, and then scientists, some of them, they performed on themselves also. So they stood in that in between the coils, and as you can see later on, Magnuson in 1911 they designed certain head coils also, and they passed the current, and they what now could be recorded not only as a EMG is one thing which can be recorded, but then what easily can be recorded is your visual sensations or phosphenes as we call it. So you feel when the electric current is passed or magnetic field is there, you feel as if a light is moving. Okay, so those are visual sensations. So that could be very easily recorded. So those were recorded, and therefore a series of experiments were done wherein uh, they gave different strengths of currents, and therefore different amount of Tesla of magnetic fields were generated, and therefore they tried to find out what is the threshold, how many turns should be there, and therefore this concept of stimulating the neural tissues or the body tissues with the help of magnetic fields came into picture, right? And then 1980, Merton and Morton, they gave the concept of transcranial electrical stimulation. So now we know that if we keep the electrodes onto the scalp, then we get different tissues, okay? And simultaneously around 1985, then the concept of transcranial magnetic stimulation came into picture, that if you place a coil and a magnetic fields are passed through that, magnetic fields are generated through that, then you can stimulate the specific areas of the brain. Okay, so that is what I'll be talking about. So there are various, so what is brain stimulation techniques is, we are using this, and before I think I missed it, that Heb, you all of have must have heard about the name of Dr. Heb, who had gave that the neurons which are wired together, they fire together. Okay, and therefore he also said that the activity dependent, there is changes in synapses. Okay, synapses, they are plastic in nature, and that is more dependent on activities. Okay, so what we are aiming through these brain stimulations technique is that we want to modulate the brain activity or the cortical excitability. Okay, so, and this could be any sort of energy. So it could be electrical current or magnetic pulses, or maybe focused ultrasound. Then two terms will generally come while you're going through literature or you're reading an article is neurostimulatory approaches. This is more referring to the electrical currents and therefore the scalp. And therefore, if you're placing the electrodes, you're more inducing directly the action potentials, okay, which I'll be talking about, because you're mostly targeting in this electrodes. Is the electrodes can be very small in size, right? And therefore it can be so small that you can directly stimulate the pyramidal cells within the cortical layers, and therefore can directly induce generation of action potentials. Whereas the neuromodulation basically refers to that you're modulating the activity, okay, the excitability of the neurons, and therefore indirectly in that case the pyramidal cells will be getting stimulated, as in case of transcranial magnetic stimulation. Okay, so but both the things can be, they are non-invasive, and therefore they can be applied through the scalp, and therefore they are operating from a range of very small circuits, like directly stimulating the pyramidal cells or through the transsynaptic connections, and therefore wider brain areas can be stimulated. So if you're stimulating the cortex, though we are modulating the activity of the cortex, but the areas which are connected to the cortex definitely will be also getting influenced. So like for Parkinson's disease, if you have to talk about, then the deficit is in more in basal ganglia or substantia nigra. But most of the studies you will see they're targeting the motor cortex, because that is, you can easily target the motor cortex on through the scalp. But then through the indirect, through the connections of motor cortex to the to the basal ganglia, you are indirectly affecting the modulating the activity of substantia nigra as well as striatum. Okay, so you can have target smaller microcircuits or to wider brain regions, and the effect has been postulated to persist for hours to days, which I will be talking about. So there are a number of non-invasive techniques which are available: transcranial direct current stimulation, electrical nerve stimulation, vagus nerve stimulation has also non coming into picture, and lot of everybody is talking of vagal nerve stimulation also, radioelectric stimulation, focused ultrasonic stimulation, and then what is my area of interest is transcranial magnetic stimulation. And then another brain stimulation technique, which is actually invasive, is DBS, which I'm sure again all of you must have heard about. If we compare the different techniques which are available as compared to the pharmacological or optogenetics and other type of techniques, then what you can see that this is spatial resolution and this is temporal resolution. So if you're talking of optogenetics and microstimulation, they are more research based techniques, and therefore they have a very good spatial or temporal resolution as compared to the drug therapies. But this is where somewhere in between our non-invasive transcranial magnetic stimulation and electrical stimulation stands. So they have an optimum spatial resolution and optimum temporal resolution, and therefore, and obviously the advantage is that these are non-invasive and therefore has been now actively taken up by a lot of as well as researchers. There are various devices which are available or can be manufactured for giving electromagnetic field stimulations. So we have a specific now a rat coil available in the market. So different companies are making these coils. But when I started my work in 2003, then these coils were not available. And then obviously I also didn't have the expertise, but with the help of Dr. Bihari at GNU, we made this magnetic field chamber, and which in the central they were able to generate uniform magnetic field. And then for number of studies which I'll be presenting today, we used actually this whole body exposure chamber. But now for last 2-3 years we have got the coil, rat coil, and we are now using this specific rat coils, okay, which is quite similar to the one which is being used in humans. So humans also have a similar coil, and therefore if now that we have this coil which is quite similar to human, we can plan the similar protocols which are used in humans, and therefore now using the rat models we can study extensively that what exactly is the mechanism and what protocols can be designed, right? Then this is another one type of chamber wherein you can only stimulate the whole head. So there are different types which are can be manufactured in rat models. So like I said, so electromagnetic induction is the principle of the TMS. So what you do is you place a coil on the scalp. Okay, this is a circular coil. There are different types of coil, and you pass an electrical current. So if you pass an electric current, then magnetic fields will be generated. And the advantage of using magnetic fields is that they can easily pass through skull. Okay, so there is no so-called of interference of the bony structures which are present. Okay, and so once they're there in the inside the tissues in the cortex, then these magnetic fields will generate currents. And if there is a currents being generated, then obviously you are altering the neuronal membrane properties. Okay, so that is how we are using this concept of electromagnetic induction to modulate the activity of the neuronal tissue. However, the question is that all the neurons may not be aligned similarly in the cortex. So it is not that all the neurons which are present in the cortex, they are perpendicular to the surface or they are parallel to the surface. So the amount of current which will be induced or the modulation that will be there will be dependent upon the orientation of the neurons. So if you can see here, there are different orientation of the neurons. Some could be at 90 degree, others could be at 0 degree from the surface. So the red color shows that what was the electrical field, and what is this electrical field, what is the angle which it is making with the neuron. So if the angle between the neuron orientation and the electrical field is 90 degree, then it is said that it is a cosine angle, and cos theta or cos 90 degree is 0. Okay, and therefore there is no current, virtually minimal amount of current will be induced in the neurons which are perpendicular to the surface. Whereas the neurons which are parallel to the surface, then the angle is 0 and therefore it is 1. So maximum amount of current will be induced in the neurons which are parallel to the surface of the scalp. And in between, then depending upon the orientations and therefore the angles, varying amount of current will be induced. So different neurons, depending on their orientation, will get modulated differently. So if you're stimulating the cortex, so this is motor cortex. So that's what I was talking. The pyramidal cell is the one which is the major output. Okay, and then the layer 4. So if we, like I said, if it is a very specific stimulation, if you use electrode, then you may be able to stimulate the pyramidal neurons. But since you're using a coil, and therefore a wider area is being stimulated, and therefore what we are doing, we are indirectly stimulating the pyramidal neurons through the trinsynaptic activation. And then these pyramidal neurons, they form the corticospinal tract and it goes to the specific muscle. Okay. And then if you're recording EMG, then you know, okay, that this intensity is sufficient to elicit a muscle contraction. Okay. Otherwise, how will you decide that how much intensity of current you have to give to the coil? So you have to have some method by which you can do recording and say, okay, so you have got a muscle twitch and therefore this should be the threshold intensity. So for doing any type of stimulation, like we do in animal models, we need to find a threshold. So in humans, we find a threshold, or even in animal models, we find a threshold like this, that you put on the motor cortex through the cortical spinal tract, then you observe, either record the EMG or visually also you can see whether there was any muscle twitching or not. And once you get a muscle twitching, then again, then you have to do 10 trials, and at least in 10 trials, out of them 5 trials, you should get an amplitude of 50 microvolts. So that is the standard procedure we use for defining the motor threshold, right? Okay, so then in transcranial magnetic stimulation, we can give either low frequencies or high frequencies, and depending upon what frequency you are giving, you can either stimulate the brain or you can inhibit the brain. And like I was saying, the effect persists for longer durations. This is because when you're giving this type of stimulation repeatedly, so you're not giving single pulse, we're giving repeated pulses, more number of pulses, maybe 1800, 2000. No, sir, it's okay. Yes, sir. So, so it either induces LTP or LTD. So if you're giving low frequency, which is less than equal to 1 hertz, then there is a decrease in synaptic efficacy because it induces LTD. Okay. So through the calcineurin pathway, there is downregulation of AMPA receptors, which results in LTD. But if you're giving high frequency, which is more than 5 hertz, that increases the synaptic strength again through calcium, but now it is CAM kinase, which results in upregulation of AMPA receptors. Then it also acts at the level of, results in phosphorylation of CRAB and other proteins, and therefore can induce synaptic plasticity. Okay. So that is the mechanism, actually has been postulated by doing number of studies and has been reported that it is through the LTP and LTD that we are altering the synaptic plasticity. And therefore we say that the effect is activity dependent and that it persists for longer durations, right? Okay, so there are different types of coils, like I was talking about. Figure of 8 coil. This is figure of 8 because it is making a 8 figure. Okay, very easy to remember. This is double cone. It has, because there it is in a cone shaped, and therefore this is double cone. These are, and then could be different sizes. This is bigger than this figure of 8 coil. And this is the rat coil which we have and now been using. But this rat coil is a circular coil. So it is just one circle. Now, what coil you're using, say if you're using a cone coil, then you are able to target a much deeper area as compared to if you're using a figure of 8 coil. So although I did say that if the magnetic fields are generated, then skull is not a barrier. But as it goes into the brain, its intensity decreases. Okay. And therefore the magnetic fields are able to go up to a maximum depth of 5 centimeter, which generally covers your cortical level. Okay. And therefore, in most of the studies, if you read about Alzheimer's, Parkinson's, and different studies, they're saying we're targeting hippocampus or we're targeting basal ganglion, then it is indirectly through the motor cortex or the prefrontal cortex, and therefore different areas are being stimulated. So depending upon what area you want to target, you stimulate the appropriate cortical area. Okay. So, and then this figure of a and double cone, like we have been working on spinal cord injury patients, and we were targeting the leg area. Now, if I think most of you are neuroscience students, if you remember the homunculus, the sensory or the motor homunculus, the legs, they go inside the central gray sulcus. Okay. So, the sagittal sulcus. So the legs, because they're going inside, dipping into the sulcus, and therefore, if you use a double cone coil, they're able to stimulate the legs. But if you want to stimulate the hand area, then it has to be more superficial, and therefore you keep it on the scalp. So you do different type of, therefore you can use different type of coils. And therefore the outcomes of TMS will be dependent upon what size and shape of coil you're using, what is the orientation of the coil, which area you're targeting, and then how much active your brain area already is. Okay, because you are modulating, and therefore if the area is already quite active, okay, and therefore, and that, like in case of schizophrenia or psychiatric disorders, generally if a person is, then you need to inhibit. So, depending upon, or in say in Parkinson's, if there is a bradykinesia, the area is, inactive, and there is a motor deficit, then you want to stimulate that area. So, depending upon what at the rest, what is the excitability of the area, you either give low frequency or high frequencies, and then how many trials, how many sessions, and what are the stimulation parameters. These all will determine the outcomes. So not only it can be used for therapeutic applications, for various psychiatric disorders, it has been FDA approved. For chronic pain, it has been FDA approved. But for various neurodegenerative, neurotraumatic, and neurodevelopment, it is still under clinical trials. And we are hoping that in near future, we will get some FDA approvals for that. But as a physiologist, neurophysiologist, I'm more interested in understanding the mechanisms, even in patients also, and therefore TMS can be used for used single pulse TMS or paired pulse TMS. There are different paradigms of TMS. They can be used to understand the mapping of cortical regions. So you can map a brain specific, say a hand region, one finger region can be mapped very crudely. It's not very specific, but you can find out that suppose there is a amputation of the limb, of the forearm, this area, then that area generally shrinks. Okay. And the other, which is adjoining areas, they take over. Okay. So that we already know. So if you are giving an intervention, then you can see that, okay, whether the area which has gone, which has shrunk or reduced in size, whether that is recovering back or not. Okay. So like, especially we are doing that in cerebral palsy. So you can do functional mapping. You can produce temporary virtual lesions, not actually lesion, but virtual lesions. And therefore you can study the role of that area in performing a task. And then various type of plastic neural networking, neural circuitry, neural plasticity type of works can also be done. So at AIMS, then we have both animal models, and we are also working on patients. And my main focus in neurotraumatic, neurodevelopmental, and neurodegenerative disorders. So I'll be just giving an overview more about neurodegenerative and something about neurotraumatic, and mostly about the animal models. Humans, this time I've decided not to talk about much about it. And so why do we need animal models? Okay. So obviously, to understand the therapeutic efficacy in this case is TMS, that whether it is able to relieve the symptoms, to what extent the symptoms can be released, how it can affect the progression of the disease, if it can reduce the halt the progression of the disease, to understand the mechanisms, and to maybe to able to standardize the dose and duration, and maybe to devise other strategies of giving stimulations. Okay. So this slide in included because, like I said, that we had customized that whole body magnetic field chamber, and therefore the coils, such coils can be built easily. So what you need, coil is basically a core which has a number of turns around it. Okay. And then so solenoid, which is an insulated copper wire, pulse generator. So you can generate different pulses, have some, so appropriate stimulation parameters can be given, sensors which can tell that how much is the intensity. But what is most important in this case is that if you're using such coils, then and you're passing electric current, they get heated up. Okay. So you have to be very careful, even if you are doing on animals or in patients, that coil, you have to have an appropriate coolant along with it. or you should have a certain covering so that the heat is not transmitted to the animal, because then it will be uncomfortable and there will be a lot of confounding factors. And sometimes there is a lot of sound also coming. So if you've seen those high voltage wires which are running from one city to another, they vibrate a lot and produce sound. So that could be another confounding factor. So these things need to be taken care of when you're designing this. So we made this whole body magnetic field chamber. So it had 4 coils. So this was basically a modified Lee Whiting coil, wherein they have proposed that if you have 4 coils and the ratio of the turns of the 4 coils is 9:4:4:9, then within the center of this chamber, you will achieve a uniform magnetic field of 17.96 micro Tesla. Okay. So then there were other designs, but depending upon the space availability and our aim, we chose to have this Lee Whiting coil, modified it, and we use this chamber for exposing the animals. So first of all, I'll talk about what we had done in Alzheimer's disease. So we made Alzheimer's, all of know, is a progressive degenerative disorder resulting in loss of memory, thinking, and others. And it's a quite prevalent. And in your future, we hope that number of cases will be very steeply increasing. And therefore, it is a disease of concern. There are a lot of FDA approved drugs which temporarily do reduce the symptoms or maybe try to reduce the dementia, but then there is no standardized drug which can restore or can revert it completely back or halt the progression to a significant effect. And therefore there are a number of clinical trials going on, and we are targeting the magnetic stimulation. There are different type of animal models for Alzheimer's disease. For spontaneous models, there are few in dogs, cats, and bears, but not in animal rats and mice. And but what we were then there are transgenic and other lesion models also, but these chemical induced models are more common. And we use streptozotocin for the reason which I'll be just coming to. Because what we need to remember in neurodegenerative disorders is that they are very progressive. It's not that one day you will find that you have Alzheimer's or one you have Parkinson's. The symptoms, there is a gradual degenerations, and therefore the symptoms develop over time. So we need to have a drug. If we want to mimic those symptoms, then we should have a drug which is producing gradual damage, and therefore over a period of time the model is developed, and therefore resulting in 60 to 70% of damage of the neurons, so that there is a cognitive deficit or if there is a motor deficit, right? So that is what is streptozotocin doing. So it's it has homology with the glucose, and therefore it is being taken up by GLUT receptors, and once inside the cell, then it can result uh, deficiency of ATP, and then it can have alterations or in the neurons or in the glial cells, and therefore resulting in neuronal death. But what is again is most important while using streptozotocin is that the key features of Alzheimer, if you know, is your senile plaque and neurofibrillary tangles. So that is how we define the Alzheimer's disease. And using this streptozotocin model, then actually it results in the formation of senile plaques and neurofibrillary tangles. So if you give streptozotocin, then it takes approximately 24 to 30 days, maybe 3 to 4 weeks for top senile plaques to appear, or and then another maybe 40 days for tangles to appear. Okay, so it is a long procedure, but then we did look for these, and I'll show you now. So the similarities between AD model and streptozotocin are quite a long list. So they do produce cognitive deficits, A beta plaque, tangles, reduce energy intake, oxidative stress, damage to cholinergic neurons, anxiety, neuroinflammation, gliosis, so and so forth. Okay. And then obviously there are others like language deficit, we don't know. Okay. Then what is the mood or attention? Some of the things which we are not able to study and not evident in animal models. So we had 4 groups. One is healthy group, sham. So we injected only the artificial CSF in lateral ventricles. So this is a intracerebroventricular injection. Then there was a sham group along with MF exposure, because there were some studies which say that if you give to a healthy subject, healthy animal, if you expose them to the magnetic fields, then there is a release of neurotransmitters, because you are basically modulating the activity. And if they are active, they will maybe actively secreting and synthesizing various type of neurotransmitters. And then magnetic fields were given to this model, and this study was done for either 30 days or 60 days. So this is the chamber which we used. So this is a standard procedure for studying the spatial memory, okay, and any cognitive deficit. So this is, I hope all of you must be aware, this is a Morris water maze, which has 4 quadrants. This is the platform here, and we give a cue, and the left animal is left quadrants. And to be able to survive, the animals have to struggle and they reach the platform, and that's how they gradually learn over a period of time. Okay, so you can use that. So what you can see in this diagram is, this is 4 days we had given the trial. Every day the animals were trained, and then gradually, so this is this line is for the AD rats. So there was a gradual learning over a period of time as the day progresses. But in this diagram, what on the right side, what you can see, the baseline data, if you see before any intervention, then all the groups had similar, this is on the y-axis, the latency to the platform. Let's suppose if you have left in first quadrant and your platform is in fourth quadrant, how much time it takes for the rat to travel from first quadrant to the fourth quadrant. So that is what is latency. So latency was very similar in the baseline when you see the baseline data. But after the surgery and intervention, this one is the AD group. So there was a significant increase in the latency to reach to the platform. So they were not able to, even after 4 days of training, when fifth day the test trial was given, the latency was quite significantly more. Okay. And this is again showing you the intergroup comparison. So in this AD group, as you can see, the latency is quite large. Okay. In AD, definitely it was slightly larger, but in comparison to the AD group, there was a significant decrease. Okay. So similar in other graphs which are depicting different parameters, how much is the time duration and other parameters. So we got similar results in other parameters. What I want you to note is, this is the heat maps and this is the track plot. So this is the control animal, if you can see. And this area, this quadrant had the platform. Okay. And therefore you can see that the animal was spending quite a lot of time on this area, and therefore the color is more prominent here. Okay. This is in the sham group where we had not given streptozotocin. Again, you can see more intensity here, but just look at the AD animals. If you look at the AD animals, then in the fourth quadrant, there is no such change in color. Okay. And the animals were more or less exploring the area, and therefore this type of behavior called as thigmotactic behavior. They were more moving towards the periphery and just exploring the area. Okay. So they were not really reaching and staying onto the platform zone. This is sham plus MF. So again, it is similar, but it is AD plus MF. If you see, yes, the exploratory activity as compared to these were definitely more, but then lot of time they were also spending in the platform zone. Okay. So we say that there was definitely much improvement in their cognitive function. They were able to recognize, stay there, but they were not 100% restoration. Okay. Then we also did avoidance memory, which is passive avoidance. So this is the step down latency. So this is basically, we used a Skinner box, which had a grid floor through which the shock was passed. And this was the wooden platform onto which the animal was kept. And then as soon the animal stepped down onto the grid floor, they were given shock. And then the animal was removed. Next day, 24 hours later, we again kept the rats onto this wooden platform. And now if they remember, then they should not step down. Okay. Or they should take more time to step down. Okay. So that is how you're assessing. Again, if you see, baseline data is similar. In AD animals, they immediately step down. So as soon as we were leaving, they stepped down. But AD plus MF group, there was a recovery, significant recovery. Okay. So that is what we have calculated in other parameters also. Then recognition memory, another test is novel object recognition task. Okay, so that's what we do. In this task, what we do, this is an open field arena. Okay, so we have two objects which are quite similar, and we allow the animal to explore the two objects for 5 minutes. After a gap of 15 minutes, Then we change one of the objects. So one of the object is a novel object. Now rats have a tendency to explore more the novel object rather than the familiar object. Okay. And therefore again we for 5 minutes we explore, we see that for how long they have explored the novel object as compared to the familiar object, and therefore we calculate the recognition index, right? So again, this was used using anime software, which I think the company is also sponsoring this event. So they were using this nose point, body point, and tail point for movement of the animal, and therefore the exploratory activity of the animal. These are the heat maps. So baseline again was similar, but as you can see in AD group, there was a significant deficit in recognition. So they were exploring both the objects more or less equally. Okay. And therefore, there were not any preference for the familiar object. Whereas in the AD group, there was a significant increase and they were able to recognize that this is a novel object. And therefore, the exploratory activity towards the novel object was quite large. Then to text the anxiety, I think most of you again must be familiar with this elevated plus maze, which has two closed arms and two open arms. Okay. So, and this is kept at a height. So what is more important is that this is kept at a height of around 40 to 50 centimeter. And if the animal comes in the open arm, they will be afraid of the height, and therefore their movement towards the open arm should be less as compared to the closed arm. They may and if the animals are more anxious, then they'll be staying more in the closed arm as compared to the open arm. Okay. So we calculated the ratio of the time spent in closed versus open arm. And again, you can see in AD group, they're spending more time in closed arm as compared to the open arm. Whereas in AD group, AD plus MF group, exposed to the MF, there was a significant decrease in this. So they were spending again more equal time or not preferentially going towards the closed arm. Okay. So this is again the heat maps of that track plots. Then we did CV staining for the hippocampus. So this is C1 area and C3 area. So this is in the normal, these are not, I know, very not good, very pictures, but this is the C1 area where we could see the viable neurons. In AD group, you can see there was a thinning of this layer, and there were a lot of neurons, apoptotic neurons, which are present there. And whereas in the MF group, we could see the viable neurons, and again, there was a restoration of the integrity of the layer. And this is more evident in CA3. You can see this layer, which is, we have drawn a dotted line around which you can see the neurons. So in AD, there was no integrity of CA3 layer, which was returning back in AD plus MF group. Then we also traced their cell bodies. So this is control soma of a pyramidal neuron. And as you can see, there are axons and dendrites. In AD, there was complete shrinkage, and then actually the neuron, and there was dendritic loss, and the nucleus was not evident. So there was an apoptotic type of neuron which was present. Whereas in MF group, you can see there was axons and dendrites, and we could see some nucleus also. Similarly, in C3 area. Then we did Golgi-Cox staining to be able to see the dendrites. Okay, so how the pruning is being taking place or not. So this is control and sham animal. Now you can see this in the AD group. Even the soma is not evident. It is shrunken so much, and you can see both apical and basal dendrites are more or less gone. Okay. Whereas in MF group, definitely it is not quite similar to sham of control, but you can appreciate a cell soma, axon, and various dendrites which are present. Okay. So that is the effect of magnetic field exposure. Then we did congruent staining for a myeloid beta plaque. And these arrows are actually indicating the presence of beta plaques in the AD group. Okay, these red dots, which are orange dots, which are present. So these are beta plaques. And then we also did staining for HP tau. And he did counting also. My PhD student work, which is the one which I'm presenting. So there was a significant increase in the expression of HP tau as compared to AD plus MF group. So with this, we summarize that streptozotocin was able to produce the model. And then if you're giving magnetic field exposure for 30 days, and then we gave for 60 days, there was a significant amelioration of the deficits. And therefore, that is maybe the proposed mechanism of the recoveries which we are seeing in Alzheimer's disease. I don't know how much time I have. Oh, okay, 5 minutes. So I'll quickly run through then. Then we also did for Parkinson's disease. So this was one MSc work wherein we thought that if we take a bottle in which there is a mild form of Parkinson's disease, so we use 6-hydroxydopamine for producing Parkinson's disease model and injected into the striatum. Uh, and this is, entering through the dopamine transporter. So this study was done for only for 7 days because of master's thesis. And then we analyzed did various scoring methods and motor deficits. So this is in the PD animal. If you can see, there was this is scoring, okay, and the 0 scoring means the normal posture, and point 0.4 means there is obvious ataxia or head tilting, and the way animal is behaving. So if you see, this is the PD rat. Their score was for 7 days being maintained around 0.3, 0.4, whereas in MF group there was a significant reduction. Okay, and then we did rotor rod test. Again, there was an improvement with giving after magnetic field exposures. We did footprint analysis. We could find only difference in base of support and not in rest of the parameters. Then we used flow cytometry to study the oxidative stress, and we found that, and this was done for striatal tissue, that there was a significant decrease in oxidative stress. So the next was that if you are giving for mild PD, what happens in severe PD? And therefore, if you're giving 6-hydroxydopamine, it takes approximately 3 weeks for the severe PD model to develop, and therefore damage of 60 to 70% of neurons. And therefore, we thought that if we have to, translate this work or make the neurologist understand then when this TMN intervention could be effective. So we did piled, and then most of the patients sometimes come when they have very severe symptoms. So we gave MF after 3 weeks, and we found that after a month there was an improvement in rotor rod test at very low speed, okay, or even at accelerating speed, but very low speeds there was an improvement. But if you go to higher speeds, then the animals were not able to maintain their balance, and there was no change in footprint analysis. And when we did the ultrastructure of striatum using electron microscopy, again there was lot of damage in all the groups, there was no change. Okay, and TH staining, again, there was no change. So we didn't find any improvement in TH change. So with this brief, I'm just presenting the brief results. What we postulated and therefore we reported is that if you're giving magnetic field exposure when the symptoms are less and therefore it is a mild version of disease, then it is most effective. But if it is a severe model, then you cannot revert it back. Okay, so you start earlier, then it is improvement. So I'll just take few more minutes, Dr. Jha, with your approval, because this is something which have been working, and this has moved to clinical trials. So I thought I'll just give a glimpse of that. So spinal cord injury is one of the major disorders, and nowadays young boys and girls, they have been driving bikes or flying kites, and therefore there are a lot of spinal cord injuries, accidents. And therefore we started with giving magnetic field stimulations, but then we thought, okay, let's be more specific and therefore stimulate only the area which is where the lesion is, and therefore we used nanotechnology, and we used iron oxide nanoparticles for that. Why iron oxide? Because if you're giving magnetic fields, the nanoparticles will get magnetized, and therefore they should have more local effects. Okay, so that was the case that we started with. So this is spinal cord injury. So I'll just skip these slides. So we have similarly a lot of groups. And so one of the major symptoms is that they are not able to control their urinary bladder. And so this is the SCA spinal cord injury group. So we showed that in this group, spontaneous recovery definitely is there, because if you remember your neuroscience, we have a parasympathetic division going to the urinary bladder as well as sympathetic. And therefore, if you're giving the lesion at the T13 area, at least the parasympathetic, which is going to the bladder, is maintained, and therefore there is a recovery of bladder, but it takes approximately 25 to 20, more than 20 days. But if you're giving an intervention only MF or the combination therapy, then it is most effective. And within 10 days, most of the animals were able to recover their bladder function. Okay. Similarly, in the locomotor function, we did BB locomotor score, which is standard used for SCI, done in an open field arena. So we did for 5 weeks. And as you can see in the first week itself, so we started the intervention 24 after giving the injury. So the intervention was very early. And therefore, you can see that there was preservation of, there was a deficit, but then at first day, there was certain locomotor activity seen in the rats. And there was a gradual improvement. And at 5 weeks, there was a significant increase. And if you're giving MF alone, also there is recovery, but then the recovery starts later. So if you're giving nanoparticles along with MF, then the recovery is faster. And if you see this damage volume, then you can see the lot of damage is there. So this is the area where we produce the complete transection. But gradually, even spinal cord injury is known to called as a neurodegenerative disorders, because gradually both in the costal and nodal direction, there's progression of the neuronal damage, and therefore there was a lot of gliosis inflammation. But if you see NP plus MF group, there was a significant reduction in the secondary damage. Okay. And so we were able to see a lot of motor neurons also present in the area of the lesion area. And then another etiology very important with spinal cord injury, that a scar is being formed. Okay, so whenever there is an injury, a scar is being formed, and therefore, this is, we did Masson trichome staining. So you can see a lot of connective tissue, which is, or collagen fibers, which are present, which is indicating scar. But if you see NP plus MF group, there was complete abolition of the scar. Scar was not there. So, and why it is important to remove the scar? Because scar will then hamper any type of axon regeneration from many of the sites. And therefore, it is very important, especially in SCI or TBI-like conditions where there is a formation of scar, that it should not allow the scar to be formed. So this was very, very significant report which we have published in 2013. And this paper got accepted within 1 month and got published, just because we showed that scarring was totally gone. And then there was also sprouting and axon regeneration, and we could see the nerves being regenerated and passing through the lesion site. Okay, so these were very significant achievements. And so keeping these achievements in mind, we got permission from DBT and funding to start the clinical research trials, and we got ethical clearance, and we have done around 15-20 patients now and getting good results. So and that's what that besides that, the protocols which we are using in patients, we are also working on rats. So we haven't left the rat work, we are still continuing with it. So exactly the same protocols we are doing in rats to further understand that what exactly must be going on to various neurotransmitters and various other mechanisms, and even to the proteins. So one of my PhD students, she is now working doing proteomics and trying to understand how the proteins are expressions is being changing. Okay, so this is my research group, and thanks to all of you for patient here. Thank you.
Thank you so much, ma'am, for the inspiring talk. Since we are running late with the time, we can have like 1 or 2 questions.
Your presentation, you basically talked about how magnetic fields basically work similar to like when we are using the effect is basically stimulating the brain neurons, right? So in all of the results where you used Alzheimer's disease, be it Parkinson's, along with magnetic field, what exactly is the magnetic field doing, which is causing it to heal? Like not heal, but right, becoming better.
So that's what I said. No, it's electromagnetic induction. It works on electromagnetic induction. So the major disadvantage of using electrical current is that it is invasive. Or if you're putting the electrodes directly on the scalp, then there could be contraction of lot of muscles, and therefore there could be headache and other issues associated with it. And therefore it is always preferred that you should have some method which has minimum side effects and is a non-invasive method. So what is being done, that if you place a coil and you pass the current, now that coil which is there, that inside of that there is not must be number of turns are there. So you're passing the current that will reduce the magnetic flux. वो physics में हम ऐसे ऐसे flux draw करते थे ना, so that is what magnetic fields are being generated. They'll pass through the skull. Now once they're in the skull, okay, so these magnetic fields should be able to induce, because these are excitable tissues, they should be able to induce certain electric current or eddy currents as we call it. And if they are there, then membrane being a has a transmembrane potential. It has a potential difference. So that potential difference will get altered. Okay, and therefore, due to the alteration of the potential difference, we hope that the channels which are present will be altered, their activity will be altered, and therefore in vitro studies it has been shown, especially for the calcium channels, that their activity is modulated, and therefore the calcium may, depending upon what frequencies and all you're giving, they may enter or they may leave. Or even the opening of the sodium potassium channels, okay, and the permeability of the membrane may be altered to a certain extent.
Okay, so we'll move to our next talk. Let's thank once again to our speaker and we'll move to the next talk.
Thank you. Thanks for.
Thank you so much, ma'am. We are really grateful. But before I disturb you again, I would ask Dr. Rishikesh to present you the memento. Tathagat?
Yeah.
We are grateful to Professor Jain for generously sharing her scientific expertise. Now equipped with the valuable knowledge from Professor Jain, we shall gracefully seek into presentation of our next speaker, Dr. Sushil Kumar Jha, Associate Professor, School of Life Sciences, Jawaharlal Nehru University, Chairman, Special Center for Systems Medicine, JNU, Member Secretary, Institutional Ethics Review Board, JNU. He is a distinguished name for studying the role of sleep in memory consolidation and the cellular and molecular mechanisms of the same. His unwavering dedication towards research has led him to receive several accolades. To name a few: Professor Baldev Singh Oration Award by Association of Physiologists and Pharmacologists of India, Scopus Young Scientist Award by Elsevier, India, Young Investigator Award by Sleep Research Society, USA, Faculty Career Development Award by American Sleep Medicine Foundation, USA, B.K. Anand Research Award by Association of Physiologists and Pharmacologists of India. Sir, we look forward for the enrichment from your field of expertise. The title of his talk is Neuroplasticity and Compensatory Mechanisms of the Brain. Sir, can we please have you? Yes, sure, sir. Data, it seems transfer is not performed. Thank you so much.
Hey, if somebody has a question for the Jan, they can think that is clear. Okay, so in the meantime, we can continue with the question and answer session with Dr. Jan. If anybody has any question, because we interrupted, yeah. Yeah, change the faculty here.
Hello, Professor Jain. Thanks for the wonderful talk. I have just two technical questions. First is that how reversible this process is. If you are stimulating neurons once, then can it be reversed after some time? You need more stimulation sessions?
Yeah, so this is a very good question. I think I didn't, took it in my studies. But since these are activity dependent facilitatory effects, okay. So, and then, so it is always that we say that whether it is Alzheimer's disease or Parkinson or any condition, it has to be associated with certain, physiotherapy or rehabilitation programs. Okay. So, like in case of, say, Alzheimer's disease, then we generally the doctors, they give them some tasks to do so that those activity which has been induced should be sustained. Okay. And if you stop using your doing any cognitive task, then it is quite possible that it may come back to its normal level, because whatever body doesn't, its body feels is redundant, then it goes back. Okay. So therefore, it is very important that when we are given TMS, it does stays for 6 months or 1 year, the effect goes on. But then for longer sustenance of the effect, you need to continue working and using your neurons so that they remain healthy and active.
Okay. And the second thing, since it's a bit non-specific stimulation you are giving to the whole region, so what it is doing to the healthy neurons, which are already functioning normal. So if we are stimulating them also, so it affects their function as well.
Yeah, so it's more or less one is definitely so it is in the healthy neurons, actually it is inducing compensatory mechanisms. So there could be more synapses which are being formed on the healthy neurons as compared to the neurons which are dying, because the neurons which are dying, what this is doing is trying to make them, make them healthy, but they may or may not come back. I mean, they may not revert back. So if you start the intervention quite early, first is that it will reduce the number of neurons which are undergoing apoptosis or necrosis. Okay. So that is one. The other is that those neurons which are already present, synaptoplasticity will be more in those neurons. So even if there is 50% damage or 20% damage, then the compensatory mechanisms will be more effective, and therefore that will try to bring back the function.
Hello, ma'am. I have a question. When we are stimulating these brain regions, if we stimulating one region, one thing is like what happen on the other part of the brain when we are stimulating the projecting neurons, whether they are going to get stimulated. And when we are talking about the memory. Are you all right? Okay. Okay. So
Are you okay? Just there.
So what I was wondering about when we are doing these Morris water maze, one of the things that have we study the population, because every mice have a different cognitive abilities. So one thing is like if one is very good at recognizing the sport even after the lesions, so they might be able to do it in very fast way. Have we done that? The susceptible and resiliency about these lesions. The other thing is when these memories are being stored, they are stored in somewhere else. Have we done the prefrontal cortex or cingulate cortex regions that we are losing some of these connections in these regions or not?
So, like you rightly said, the storage is in prefrontal cortex. So in Alzheimer's, actually, we are stimulating the prefrontal cortex, not the motor cortex. Whereas in Parkinson's, because the primary symptom is motor cortex, and therefore you're the motor deficit, and therefore you're stimulating motor cortex. Okay. So nowadays, therefore, depending upon what disease you're targeting, you may be stimulating different areas. Okay. And therefore, in certain conditions, when they found that the person was not able to maintain, there was cerebellar ataxia, then they found that nowadays they're stimulating cerebellum. Okay. So therefore, there is different areas are being stimulated, right? About the mice, there are different effects of cognitive abilities. So that's what I'd shown earlier, that we take a baseline value and we see that all the mice, they're falling in a normal range, and therefore there is a uniform distribution, so called. And otherwise, what we do, if we find that there is some mice which are very good and some bad, Then we take a percentage change or normalize the baseline values, and then we see what is the effect. So that is more statistically, we try to rule out those changes. Okay.
Okay, so we'll continue with our next talk.
Sorry for the interruption, because we have to keep time again. But she will be here. I hope she will be done. Once will be, both of you, so that people can talk. Yeah, you just start with.
Yes, sir.
Good morning, and thank you so much, Dr. Madik, for inviting me. And though it's a very nice introduction, but I will not present data on sleep. I understand this is a workshop. So I will just put forward some things that is very exciting, at least to me, what has happened in neuroscience and what is going on recently. And Professor Jain has set my life easy to give, she gave some important aspect, how neuron recovers. At least with the magnetic stimulation, she showed that there's a recovery. And what we know, we have no fixed number of neurons in the brain, and damage is damage. It doesn't recover. So I will be talking about that. So if you look at neuroplasticity, the way it is defined is the brain's ability to recognize itself by forming new neuronal connections throughout the life. Means it says throughout the life, if there is some damage, there will be some recovery. And it can happen at many places. For example, if you see here, so this is written neuronal plasticity. This is synaptic plasticity. Here it is. And what some examples she said, like she has given, and also the pruning, forming of new dendrites. So these kind of dynamic changes are taking place every time. But the question is, where? Is it in the entire circuitry, entire brain, or a specific area? That we have to understand. And if you look at some of the salient features of synaptic neuronal plasticity, means adaptability, means it gives the brain a power to adapt the changes. Suppose we learn something, what do you expect? What are the changes that may occur in your brain? And how much time it will take? Anyone has any idea? If you learn some new task properly, what are the changes that you expect in your brain? And how much time it takes? That's you're talking about problem in retrieval. But what if the problem about the information is stored properly? During examination time, you learn so many questions. When you go and write your examination, it pops up very easily because 3 hours you have to. So what are the changes you expect in your brain? That is very dynamic question. I will give you some examples on that. And types of plasticity is, here I am mentioning only functional and structural. I'll give you example. And the third one is compensatory. So I will not go in that detail. What has started, if you go back the history, so the two crazy guy, Bliss and Lomo, they were doing some experiments on rabbit brain, and their job was to stimulate. And what they did, before that, some information was available. Anderson's lab were working on that, and they were inducing this long-term potentiation, what Professor Jain has taken the name, but they were lasting only for a few seconds. And this crazy guy, Lomo and Bliss, what they did, they stimulated with a tetanic stimulation more than 100 hertz. And what they found, this potentiation was lasting, what is here is 20 minutes. And that was very, very exciting, that you do something in the circuitry in vivo, and then potentiation lasted for 20 minutes. Later on, again, the same guy did it lasted for 60 minutes. So then this neuronal plasticity field was becoming more and more fascinated that what is happening if you stimulate the neuron with high frequency, what is happening at the memory level, why they are potentiated and why they are lasted. Even you see this effect was lasted for how many days? 360 days. But this was all artificial. Means you have to stimulate electrically
magnetically, what she gave the example. But what happens, and then it was correlated with learning. When we learn, after learning, this kind of potentiation, does it happen? And it was shown by this group, Whitlock group in 2006. Yes, if we learn some task, this membrane is getting potentiated, neuron and firing rate is increased. The same neuron was firing 2 hertz, 3 hertz. Now, after learning, when they acquired the information, those neurons are firing with the higher frequency. And what they did, they put this array of electrode into the hippocampus. This is the dorsal hippocampus. And out of 8, they found only 2 neurons. 6, there was no response. 2 neurons was potentiated. People asked question and they commented, out of 8, only 2 potentiated? Then the answer was, it is not that every neuron is going to get potentiated. What you learn, where it is getting stored. So you have to have an engram cell, concept of engram cell. So the neurons, those are involved with acquisition of information, those are called engram cell, and those neurons will be only potentiated, not the all other 100 neurons. And this is what the first time it was shown that learning, what you can see all this heat map here, they are the activated response. That was considered to be the functional plasticity because the function of neurons had changed. And it happens within few hours. You learn, you get the information consolidated, you don't need more than 5-6 hours. And your neuronal properties change. And that is why we say we never use same brain twice. Do you agree or you don't agree? You use the same brain twice. Now at the structural level, this is my work, but what has been done, that is called ocular dominance plasticity. And this plasticity was discovered by Hubel and Wiesel in 70s. They got Nobel Prize for that. So what it means the neuron, the eyes, the 2 eyes we have, left and right, and the visual cortex, the information from both the eyes goes into the visual cortex. But there are certain columns, those columns are called ocular dominance column. Means those columns where the right eye will be more dominated and the neighboring column maybe left eye may be more dominating. So the right eye connection with the few groups of neurons, left eye connection with a few groups of neurons. So right and left columns are there, and those are called which eye is dominating, ocular dominance column. So what experiment we did, this is what it has been shown, this right eye and left eye, the green and red. So there is a proper presentation of these neurons, right eye, left eye, left eye, and there are few columns, yellow one, they are binocular. Now what you do, what do you think? If the child is born with one eye cataract, what will happen to that child? Huh? Right. If it is not removed, right, okay. And newborn babies, surgery, who's going to do that? So there is a critical period. Within that critical period, we have to remove that cataract. Otherwise, the child will be blind to that eye lifelong. So this was done in kitten. And what the experiment we performed, so this what you can see, this cat is over here. So the one eye is blocked, right eye. And after keeping this right eye blocked for 6 hours, that is called monocular deprivation, what we did, we allowed them to recover for next 6 hours. So one was kept awake in the dark and one was allowed to sleep. And then other manipulation, 6 hour closing one eye in kitten and allow them to sleep. It's a plastic changes. And compared to the guy, that animal kitten, those were not sleep deprived kept for another 6 hours. So sleeping guy where this entire column got shifted towards the open eye. And the sleep deprived one did not. So that shows how this plasticity change occurs. So 6 hours acquisition, 6 hours of sleep, entire circuitry got shifted towards the open eye. And this is what the result you can see here. This is the control one where the both eyes are representing. This is the closed eye. Now in this one, entire neurons are responsive to the open eye, not with the closed eye. So this eye was shifted. And it is not working over here. So, and now you can see the example. This is the right eye and left eye. So the right eye was closed and left eye was open. And this is what you see is the response of a neuron, right eye and left eye. So this is a left eye neuron. All these spikes are action potential of a visual cortex neuron. So this is left eye neuron. Left eye is open. Do we think any change? We don't expect any change. This is a right eye neuron in the baseline condition. More firing with the right eye, less with left eye. More firing with right eye, less with the left eye. Now you see when we close that eye and allow them to sleep, now these neurons are shifting from right eye to left eye. The neurons, those were initially connected with the right eye, now after closing one eye in kitten and allowed them to sleep, in 12 hours, it got shifted. That is the question I asked. When you learn something, how much time you expect that something will change? Answer here is 12 hours. And when that shifting occurred, it occurred when animal was sleeping, means in non-REM and REM sleep, not in wakefulness. You can see the baseline condition, REM sleep, their firing rate, and in post-MD sleep, the firing rate has changed, means during sleep, the neurons fired more, and that allowed them to shift towards the left eye. So this is, we call it a structural plasticity. In 12 hours, so you have to be here. Okay, thank you. Sorry, I will not move you on here. So 12 hours is required for the structural change. A new connection should be formed, and then neurons got shifted to the another eye. And this is what the compensatory plasticity I'm talking about, the new formula, a new name that I'm just trying to put forward. So generally what happens, what even Professor Jain has gave some example, that this is the normal neuron connectivity, presynaptic and postsynaptic. When you do anterograde some kind of lesion, damage, so what happens, this part is damaged, axon is damaged, and this will doesn't allow this neuron to grow. So anterograde, this neuron will also die. Retrograde, you have to move to that side. Retrograde means if you put something and that will go retrograde, this cell body will also die. And then it is called dying back. So what will happen? When this die, this will also die. So this kind of mechanisms are involved with neuronal degeneration. And Wallerian degeneration is also an example. You must have heard about it. But this is example of peripheral nervous system. It doesn't work in central nervous system. Central nervous system axons do not grow, ideally. But Professor Malik will must be knowing, and he will love to know, because I know he loves adrenergic neurons. So adrenergic neurons or any aminergic neurons has the power in the brain to regenerate without any assistance. And the examples are here. So what has been done here, this is called CCI, means control cortical impact. So there's the impactor, which is like with some weight, as a machine, you can damage the cortical area. And what you can see in here, this damage area, control one and the damage area. So once this CCI is used, 1 week post CCI, this is the damage. And here what you can see, this is 1 month and this is 3 months. And what you see, the green lines, these are our adrenergic fibers, pH positive fibers, where in the cortical layers, I think this is the 1, 2, or 3, some layer 1, layer 2 and 3, layer 5. So this is the normal condition. This is after injury. And you can see this is after, this is CCI, anterior part of the injury, posterior part of the injury. And you can see after 3 weeks, when these adrenergic fibers were damaged, now it is recovering. So adrenergic fibers, serotonergic fibers, dopaminergic we do not know, but these two fibers have a capability without any assistance, this can grow, regenerate. What happens to others? They can also grow, but they need assistance. Some molecular event must be triggered to regenerate. They normally, if that is not triggered, they will not regrow. So what happens if the fibers are damaged in the brain? This is a one weird experiment has been done, and even who has discovered this is called lobotomy. Got Nobel Prize in 55, but in 74, US banned it. It is illegal to perform this experiment, and that is what is called cortical lobotomy. What you can see here, this guy, doctor is looking at, and the person who survived is called Howard Dale. So what was done, initially whosoever has proposed, a Portuguese guy, I will not name it because he got Nobel Prize, was shooting it from here, making a hole and putting an ice pick, you know, this ice block that he used to break it. So he was putting ice pick through here and then damaging the fibers that was connecting to the frontal cortex. And he was performing this experiment in maniac patient, because maniac hyperactive patient, when he was performing this kind of surgery, that surgery was making them to become cool down. And he was thinking it was because the frontal cortex had some hyperactivity, and if you slow down that hyperactivity, they will live a normal life. And I'm just forgetting the name of this doctor, who's, you can see in this picture. He was performing 25 to 30 surgeries every day, and in total 3,500 surgery he performed. And one of the survivor is Howard Dale. His brain, you can see in MRI, this part is almost damaged. And no one was really recovering, but it was because the frontal cortex, the neurons were dying. So most of the patient died, and because no one cared, and it was done between 46 to 74. Howard Dale was the one lucky guy. He challenged the world, because after this much of frontal lobe damage, anyone can say what is frontal lobe does? Huh? Basic cognitive tasks, even the decision making. He recovered and he became a bus driver, a school bus driver, after age of 45. But the surgery was performed when he was 12 years old. Because 4 years onward, he has a schizophrenic tendency, and his stepmother didn't like him. So she put him to the doctor and said, perform this lobotomy. And that was performed, and 12 years old, that time he did. At the age of 45, he became a bus driver, and he was giving training to bus driver. And then he challenged the world, the frontal lobe is totally gone, how he is taking decision while driving bus. So that shows the compensatory mechanism. Some other parts of the brain must have compensated in this Howard Duffy. Otherwise, he will not be able to drive a bus. That was the example when the brain was damaged. But what if the peripheral, some part is damaged? What will happen to the brain? And one example is phantom limb. How many of you have heard about that? What happens in phantom limb? Okay, very nice. So yes, if the part is amputated, that limb is not there, hand is not there, leg is not there. And it is there, the data says that 90% patient amputed has this kind of sensation, the part is still there. As if he's moving his finger. There's no finger. And he also feels pain. So there are two examples. One is PLS, that is called phantom limb sensation, and another is called phantom limb pain. And it is there, 77%, 78% patient reports phantom limb pain. And PLS is common in that. And then there is other terminology is called RLP, that is called the residual limb pain. So obviously there is a part that is left over will have pain also. Anyone if have not seen, because Ramachandran is the person who has done a pioneer work in phantom limb, and this is a small video you can watch. I will be stepping this corner.
One of Ramachandran's first patients was Derek Steen.
13 years ago, I was involved in a motorcycle accident, and I pulled the nerves out of my spinal cord up in my neck. They told my parents directly that I would never use my arm again. About 7 years ago, I was reading through the classifieds, and I saw an ad in there, amputees wanted. I thought it was a joke. It's just basically connecting the club to the ball. So I called the number, and it was Dr. Ramachandran.
Today, Derek is teaching Ramachandran how to play golf. But several years ago, Derek made a crucial contribution to Ramachandran's pioneering work in brain science.
Yes, that was amazing.
After my surgery, I sat up in the bed and still felt the arm there, still felt everything there. And I'm looking down and I'm seeing nothing. It was pretty bizarre. The more I thought about it, the more it hurt. The more it hurt, the more I thought about it. So it was like, it was never ending. I mean, I'd break out in a cold sweat and turn pale just standing here talking to you because the pain would hit so bad.
So Derek was his first patient, and what he reported, this is RLS, means residual limb, RLP, residual limb pain, as well as phantom limb pain. So the reason we do not know yet, but what happens, you must have heard about in your textbook, this is called denervation hypersensitivity. So if the muscle that is innervating the muscle, and if this is an innervated muscle, normal muscle, this is an end plate region. So acetylcholinergic innervation is there. So if you stimulate, you will get action potential only in the end plate region, not away from that area. But if you denervate, the connection to the muscle is denervated, the same end plate region, now you see more or more away from the area, end plate region membrane, they are now responding to acetylcholine. They are generating action potential. So what does it mean? Means there are more and more receptors are recruited away from the end plate region. It happens, it is a very normal phenomenon. And what happens? These are the receptor recruitment. It's not the normal receptor that is getting recruited. Some embryonic acetylcholine receptors are getting recruited. And that is how the more and more area will be responding. And this is a safety device to salvage the damage. Because you need muscle activity, because it has been shown muscle activity, neuronal activity is required to regenerate the fiber. So in phantom limb, what has been found, this neuroma formation has been found, and they are very, very sensitive. So slightly you touch, people will feel pain because of the hypersensitivity. Have you ever experienced burn sensation? If you burn your finger, and if something goes close by, not even touching, you feel pain. So that is what the hypersensitivity it means. And also what happens, there's like when there is a damage, some neurons will make more connection, making it more susceptible to the perception. And in this case, what happens in the phantom limb, because this part is missing, now the sensory information that is going to the spinal cord, they are not sending that information. So those neurons now will try to approach the nearby neurons, and by mistake, it has been found, it approaches to the pain alpha, A alpha, and C fibers. So they make approach to those pain fibers. And that is how the pain sensation they get, most likely. So this is the sum of the statement that the person who is feeling, like suppose a person who was tetraplegic, and see how.
Even though I am totally paralyzed from the neck down, I still have a feeling of being in contact with every part of my body. Because of the painful itching, I know where my legs are, and through the pain I can feel my knees and toes.
I was lying in bed here at the hospital and feeling really bad. All day I felt as if the nail on one toe was growing into the adjacent toe. My mother was visiting, and I asked her if she could take a look and see what was happening. She couldn't see anything wrong. Another day, I woke up with the same feeling. The pain had kept me up most of the night.
The tetraplegic are those paralytic patients whose entire body, means down the neck or 4 limbs, are paralyzed. And this paraplegic, their only lower limb is paralyzed. So in this phantom limb, this is a learned paralysis. So one, like another.
It's frozen, doctor. It's in a block of cement. If I can move my leg, it might help alleviate the pain.
This patient is feeling that it is still there, but it is frozen. Now it's unable to move. There's no leg, no hand. So what happens means this is what Ramachandran also proposed. This is a miscommunication between the sensory and motor cortex. And I'll give you an example how sensory and motor cortex behave. Means if you think, motor cortex will respond. And it is called brain-computer interface. So the electrodes are put on the frontal cortex, in the sensory area, in the motor area, and the patient, what they are thinking, they are all paraplegic. So what they are thinking, the motor cortex is giving command to the computer, and computer is performing that job. That kind of robotics has been made, and you must be knowing that Neuralinks, what Elon Musk is doing. So he's now exploring those areas, how to make use of the sensory system to perform motor cortex using the brain-computer interface. This video will tell you what happens in the phantom patient.
Only would he finally understand his phantom pain, he would also get to the bottom of a mysterious sensation he felt while shaving.
When I first started shaving after my surgery, I would feel my absent hand start to hurt and tingle whenever I shaved this left side of my face.
Meeting Derek was important for Ramachandran because the explanation he came up with would rock the world of neuroscience.
How about that?
That's just my arm.
The first thing Ramachandran did was to invite Derek to his lab for a simple test.
Derek, I want to touch different parts of your body, and I just want you to tell me what you feel and where you experience the sensation, okay?
Okay.
Close your eyes.
Could feel that on my forehead?
Anything anywhere else?
No.
Okay.
It's on my nose.
Okay.
My chest.
Your chest, okay.
I can feel that on my cheek, and I can feel rubbing on the phantom left hand.
On the phantom left hand, in addition to your cheek, I'm going to run the Q-tip across your jaw and see what happens.
I can feel a Q-tip on my cheek, and I can feel a stroking sensation across the phantom hand.
You actually feel it stroking across your phantom hand, across the palm. So here is a medical mystery of sorts. Why does this happen? Why would a person, when you touch his face...
What do you think? Like he was touching his left cheek and having sensation in the phantom hand. What do you think?
It's a normal response for him to use now. Now, what Professor Jain has already shown who will this. Okay, so what happens? What she told, the leg portion is there in this subside, right? Hand region is there in this portion of the cortex. The sensory cortex receives the hand portion in this region. When there's the amputated one, so this area is not receiving any information. And the next area is cheek. The cheek fibers, neurons, projected towards the area that was connected to the hand and finger. So made new connection to that area. And reminded, this is adult patient. Howard Duffy was 12 years old. And their compensation, because early life, if something happens, the compensation is possible. But this is again the adult one, where it shows that there is a projection from the cheek area towards the, and the MRI study has also been shown that it has been connected, because you see here MRI one, this is the normal one, this is the amputated person with no pain, but you see pain, phantom limb brain, where the connections are made to the another area in the cortex. So this shows that pain is so dynamic, always they find some compensatory mechanism to compensate the loss of pain. So do that. So with this idea, we have done some experiment in our lab. Deepika, she did this experiment, and Deepika is currently with Professor Jain nowadays. And here what she has done in her PhD work, she has damaged the dorsal hippocampus here with hemodynamic insight. 5 minutes, okay. Damage this dorsal hippocampus here. This is a normal one, and she is performing some task that is called contextual fear conditioning, and it is hippocampal dependent. So when the hippocampus is damaged, contextual fear conditioning was lost, abolished. She was not getting any breathing response. That is the hallmark of that. So you can see here, this is the normal one, baseline, and this is the compensatory, damaged one. There is hardly any breathing response. But then what she found with multiple trials, and then she allowed 3 days animal to recover after the damage, and she found after 3 days that with multiple trials, animal is showing breathing response, means connection was compensated. And where that happened, that not in the hippocampus, because hippocampus is totally gone, but in the where? In the prefrontal cortex. So this is the prefrontal cortex area. If you block this area, the activity of this area, then the compensation is very lost. So compensation is taking place in the prefrontal cortex for this kind of step. This is what the person breathing in this area. Where this area was blocked, nothing happened. And since this lab is a sleep lab, so during 3 days, if you keep the animal sleep deprived for some 54 hours and allow them to recover 18 hours, again the compensation is lost. So sleep also plays an important role in this induction of compensatory mechanism. And what is happening in the prefrontal cortex? This is what Golgi state. You can see the MPFC neurons are making much more arborized neurons. There's much more arborization. They are spreading and making new connections, and we do not know where that is happening. Since I have only 5 minutes, so I don't know. Maybe I'll just go with, I skip one. So what do you think this picture says? The picture of my daughter. What do you think? Is it real or illusion? No comment? Okay. What is happening? What is there?
Based on the arrows, we are
Is there any displacement? No displacement, but gives you feeling that it is moving left, right, up, down, right? There are neurons in the brain, especially in the prefrontal cortex. So if you see the real report, means whether the orbit was moving left or right, these neurons are responsive. And the illusion which is not really moving
giving you feeling of illusion that it is moving left. These neurons are firing much more robust. So we have a neuron that is related to illusion. Now, what is the therapy of mirror therapy? Phantom limb. There was a mirror therapy, and mirror therapy means you are putting your brain. Limb is not there, but in the mirror you see and you act. Right? How that happens? Have you heard about mirror neurons? What is that?
When you see an observation, the brain itself tries to copy that.
So here is the answer why mirror therapy is working. So what is mirror neurons? If the monkey is grabbing a banana, this is a firing ray, and a monkey is watching the man is also grabbing a banana, their neurons are also firing. Means what you see, what you do, act and see, the neurons are there. This is called mirror therapy. And with the mirror therapy, this
Brian Wagner is one of the
skip that. So with mirror therapy, mirror is used to cure phantom limbs. So means you can also mismatch the misalignment formation of the neuron with this kind of manipulation. So that shows how plastic is our brain. So it has a compensatory mechanism. It has all these the mechanism that we have to understand and how it works. I have many questions, and what I do because of all these heroes in the lab. So thank you so much.
Thank you so much for inspiring talk, sir. So session is open for the discussion. We can have few questions.
So this is Priyanshi Tiwari from AINN. So actually I have a question. I've had a very I've had experiences with people who have dealt with phantom pain. So I've noticed that they have mostly complained about feeling intense phantom pain during temperature change, especially in cold. So I've never been able to understand why heat kind of eases the pain out and the cold intensifies it a lot. So if that would be
So that was a residual limb pain or a phantom limb pain?
Phantom limb pain.
Phantom limb pain. I don't work on this. I have just copied this thing and I just presented to make it more exciting. But what I feel that seasonal change or the temperature had an effect, and especially if that is related to your past memory. Unless and until it is not possible.
Thank you, sir.
Okay, that's great. Thank you.
Thank you.
Thank you.
So after such an enthusiastic exchange of thoughts, I would request Dr. Rishikesh sir to present sir with his memento. Having been enlightened by Dr. Jha, we're ready to proceed for a small 5 minutes tea break, which would be followed by the laboratory sessions that will last till 1:15. So we would now request all the esteemed speakers, the participants, and the faculties present to head for a small tea break. Thank you so much.